Botany, University of Calcutta. Without further ado, let me request Professor UC Lavanya, connecting with us from Lucknow. He is the editor-in-chief of the Nucleus and the GB member of ASFC. Over to Professor Lavanya for the opening remarks. Yes, Professor Lavanya, can I request you to unmute yourself, or can I request Mr. Akash Model to unmute Professor Lavanya so that he can speak?
Okay, now I think I'm now audible.
Yes, sir, we can hear you.
Yes. So good afternoon to all of you, and a very warm welcome to Professor Schubert, who is joining online from Gattislaven, and to the participants present in the ARCHANA Memorial Hall, and to those joining online. On this 7th Professor Archana Sharma and Professor A.K. Sharma Memorial Oration 2024. As you know, this oration is being organized by the Archana Sharma Foundation of Calcutta, in brief, ASFC. The ASFC was founded by Professor Arun Kumar Sharma on 16 September 2009 in memory of Professor Mrs. Archana Sharma in the Department of Botany, University of Calcutta at Calcutta. To tell you a brief, the major objectives of the foundation are to publish the journal Nucleus for the benefit of the students, teachers, and research workers interested in the study and dissemination of knowledge within the scope of the journal, and to arrange and organize lectures, debates, discussions, seminars for the exchange and promotion of knowledge, and to help poor and needy students to all communities for the purpose of studies. This is very, very important aspect, last one, which Professor Sharma very much wanted, to help the poor and needy students. On the request of General Body of ASFC made in 2017, Professor A.K. Sharma agreed to initiate a memorial oration in honor of Mrs. Archana Sharma. Unfortunately, in the meantime, Professor A.K. Sharma left this material world. Consequently, the successor president of the ASFC, Professor Pakrasi, suggested to rename the oration as Archana Sharma and A.K. Sharma Memorial Oration. As such, the first oration was organized in 2018. Since then, the foundation has been regularly organizing the oration as an annual event. This memorial oration is being delivered by distinguished biologists. 6 orations have since been delivered in the past, and this is the 7th oration in the series to be delivered by Professor Ingo Schubert, IPK, Gattislaven, Germany. To Professor Schubert, he has been long associated with Professor Archana Sharma and A.K. Sharma. On my request and on his own initiation, Professor A.K. Sharma invited him to join the International Advisory Board of the journal Nucleus 2010. Since then, he has been helping the journal in various capacities and have also contributed articles in different occasions, including the latest one, The Post-Cellular Biotic Evolution in Nutshell, for the A.K. Sharma birth center issue of the journal. This article has already been published online and shall be included in 2024 issue of the journal. Thank you very much, Professor Schubert, for concluding the article for this memorable issue. With this background, may I request Professor Rita Kundu, HOD Botany, University of Calcutta, to do the speaker of the day and conduct the proceedings. Thank you very much, and warm welcome, Professor Schubert.
Thank
Rita,
you so
please.
much, Professor Lavanya. Can I now request the head, Department of Botany, University of Calcutta, and the Secretary, ARCHANA Sharma Foundation of Calcutta, Professor Rita Kundu, to introduce the honorable speaker and take over the proceedings, and after that, invite him for the talk.
Good morning, everybody. I'm Professor Rita Kundu, head of the department and acting as the secretary of ARCHANA Sharma Foundation of Calcutta. Now, I would like to introduce the audience with our honorable speaker, Professor Ingo Schubert. I'm really very privileged to introduce him. He is presently associated with the Leibniz Institute of Plant Genetics and Crop Plant Science Research, Department of Cytogenetics and Genome Analysis, Gattungsleben, Germany. He was born in 1947 and then studied in the University of Greifswald from 1965 to 70, did his graduation as biologist in the field of zoology. From 1970 to 1990, he worked as scientific co-worker at the Central Institute of Genetics and Crop Plant Research at Gattungsleben and worked on chromosomal mutagenesis in plants for his doctoral work and awarded the PhD degree in 1975. Later, in 1982, he received Doctor in Science thesis on chromosome structure, cell cycle, and origination of chromosome aberration in plants. From 1990 to 91, he was head of the cytogenetics division of the Institute of Genetics and Crop Plant Research at Gattungsleben. In 1992, he received Dr. Habil at the Martin Luther University Halle, Wittenberg. From 1992 to 2012, he acted as the head of the Department of Cytogenetics and member of the Board of Directors of the Institute of Plant Genetics and Crop Plant Research at Gattungsleben. Since 2013, he is working as guest researcher at the IPK. And his main research activities are cytogenetics, chromosome structure, karyotype evolution, chromosome mutagenesis, and comparative cytogenomics. So I would now like to request Professor Schubert to deliver his lecture. Thank you very much.
Yeah. Good afternoon, ladies and gentlemen, dear friends and colleagues. And thank you very much to Professor Lavanya and Professor Kundu for kind introduction. It is a great honor and pleasure to me to deliver the memorial lecture for Professors ARCHANA and A.K. Sharma, invited kindly by the ARCHANA Sharma Foundation and the Department of Botany of the University of Calcutta. So again, thank you very much for inviting me to have this talk in the year of the centennial of Professor A.K. Sharma. Yeah, I decided to talk to you about the work of my group and my colleagues since more than half a century at the Institute in Gattungsleben, of which you see a bird's view on this first slide here. It is a rather remote area. I think Calcutta looks a bit different. And it is exactly in the middle point of Germany. So I will deal in about a topic which moves me since several decades, namely how DNA double-strand breaks fool genome and karyotype evolution. So, double-strand breaks are ubiquitous. Can you hear me? Yeah. Double-strand breaks are ubiquitous, frequent, and hazardous. They can be caused directly by ionizing irradiation from environment or by endonucleases, or indirectly via interfering of excision repair and replication gaps. If they are unrepaired, they are lethal for dividing cells due to the loss of acentric fragments and the terminal instability of centric fragments. They are a prerequisite for the insertion of transposable elements, and they can be repaired via alternative pathways. All of these pathways can be either error-free or error-prone. The evolutionary consequences of misrepair of double-strand breaks are genome, that means chromosome rearrangements, as for instance inversions, translocations, and transpositions, and they can alter the genome size via deletions and or insertions. Here is a short demonstration of the potential repair pathways and their molecular consequences. So double-strand breaks, if unrepaired, are lethal for dividing cells. If they can be ligated directly, they restore the pre-break situation and have no genetic consequences. However, there are several repair pathways differ in the way of end processing of the breaks and the use of microhomology for ligation. The conservative non-homologous end joining causes variable sizes of deletions and or insertions below usually 3 base pairs. The so-called alternative or microhomology mediated end joining causes again sizes of deletions of variable sizes and or insertions larger than 3 base pairs. Another way of end joining is single-strand annealing between flanking repetitive sequences. They usually cause pretty large deletions and no insertions. If, however, the ends of different double-strand breaks are ligated to each other, then we obtain a chromosomal aberration. Another group of repair depends largely on homology between the broken sites and a template region. After end resection, the overhanging ends of the breaks invade an undamaged homologous double helix and resynthesize according to the template sequence. This figure called double holiday structure can then be resolved in this way, that what is called synthesis-dependent strand annealing and genetically results in a gene conversion with partial loss of heterozygosity. However, if the resolution is in a right angle with topoisomerases cutting the double holiday structure here and here, for instance, then the consequence is an exchange of the flanking regions. The resolution of these holiday junctions then, depending on what was the template strand, can either result in a sister chromatid exchange, a crossover, or a chromosomal rearrangement, as I will show you soon. Double-strand breaks are dangerous, as I told them, for all DNA inheritable material, also in prokaryotes. Therefore, corresponding repair mechanisms were already evolved in prokaryotes. However, prokaryotes have circular genomes, and if this would have dispersed repetitive sequences, of which one is broken and uses the other one as repair template, this would destroy the circular genome into two circles. Therefore, the circular prokaryotic genomes are relatively small and mostly free of interspersed repeats. And only the linear chromosomes of the eukaryotes, which ends are protected by telomeres to stabilize them, they can tolerate dispersed repeats and therefore can tremendously grow in size. Here I show you what happens on the level of chromosomes if the DNA repair does not restore the pre-break situation. So, for instance, if one chromatid is broken and uses the unbroken chromatid as a repair template, this will result in a sister chromatid exchange, which has otherwise no genetic consequences, usually. If a homologous allelic sequence is used as template for repair the break, then we get during meiosis a crossover, or during mitosis a homologous symmetric translocation. If, however, the repair template comes from non-homologous, from a heterologous chromosome, or if both are broken and the break ends are linked to each other, then we get a symmetric exchange between the heterologs as a heterologous translocations. So this happens if the proximal and the distal ends are ligated over cross. If, however, in the same situation, the proximal end of one break is ligated to the proximal of the other, and the two distal ends are also misligated, then we get a dicentric chromatid, which is unstable due to bridge formation in the next mitotic division, and the acentric fragment, which cannot segregate properly and gets lost. If two breaks within one chromosome are ligated with the ends proximal to distal and distal to proximal, then we get a 180 degrees inversion. And when the breakpoints are at different distance from the centromere, this changes the morphology of the chromosome. If, again, two breaks of the same chromosome interact with each other in the way that the two proximal ends fuse and the two distal ends fuse, then we get an acentric fragment and a centric ring fragment. If this ring fragment is replicated and after replication makes a sister chromatid exchange, the result is a double-sized dicentric ring which forms a double bridge and a subsequent mitosis and is therefore unstable. However, also disploid chromosome number alterations can be caused by misrepair of double-strand breaks via reciprocal translocations. For instance, if a meta or submetacentric break has a chromosome has a terminal break, and an acrocentric or telocentric chromosome has a break near its centric end, then the result is a large monocentric chromosomes combining most of these two and a small centric fragment. If, however, the break would be in the short arm of the acrocentric, we would get a dicentric chromosome with closely adjacent centromeres and a correspondingly acentric fragment. If the second partner is a metacentric, then the resulting chromosome would be a dicentric with large difference between centromeres. This would be unstable if not one of these centromeres become inactivated. Sometimes this is called end-to-end fusion. Also, I warn you, chromosomes never fuse because their telomeric structures prevent them from fusion. The so-called nested fusion is also a reciprocal translocation, which goes back a minimum of 3 break events. 1 in the centromere of the recipient chromosome and 2 at the terminal end of the donor chromosome, which again result in a large metacentric and 1 or 2 acentric fragments. And this split centromere is usually inactive. So, however, if 1 break more occurs, and these are flanking the centromere of the recipient chromosome, then a monocentric, metacentric combines most part of these 2 chromosomes, and 1 or 2, and a centric fragment with the centromere and these 2 terminal regions occurs. If, however, 2 breaks occur in a telocentric chromosome at the centric ends, then they can be ligated into a metacentric one and an acentric fragment. If the metacentric still contains telomeric sequences of 1 or both of these chromosomes, and a break occurs within this telomeric sequence region, then this event is directly reversible, resulting again 2 stable telocentrics. So the small products of all these events, which are characterized by an asterisk, are usually lost during meiosis, provided there is no selection pressure on them due to the harboring of an essential gene. I will show you that later on. Another way of changing the chromosome number is the simultaneous ascending and descending disploidy. This is a mechanism which 40 years ago was experimentally proven in our lab on the field bean with Faber. So in principle, this goes back on 2 translocations with 3 chromosomes involved and 1 chromosome involved in both of them. So the translocation number 1 exchanges this arm versus this arm and results in this karyotype. And the translocation number 2 brings this arm from the metacentric exchange to the short arm of this acrocentric and yields this karyotype. If both karyotypes are combined in a heterozygous condition, the individual will form during meiosis a hexavalent with pairing the homologous chromosome arms. Usually, the segregation from this hexavalent yields the parental translocations. However, in a few cases, a missegregation may occur to a few percent, in which the 4 acrocentric chromosomes migrate into one pole and the 2 metacentrics to the other pole. This means we get a hyperploid gamete and a hypoploid gamete. The hyperploid gamete reveals small duplication regions as indicated by these bars, and the hypoploid ones is lacking the corresponding regions. And the viability of this type of gamete depends on whether or not in the deleted regions are essential genes. Another way of karyotypic changes via homologous chromosome translocations are the so-called secondary rearrangements, which we detected also several decades ago in the Faber bean. And it is based on meiotic crossing over between translocation chromosomes. And again, we assume that a karyotype, that two karyotypes with two different translocations and one chromosome involved in both of them occur. The translocation number 1 exchanges this arm versus this arm and yields this karyotype. The translocation number 2 exchanges these unequal parts of the long arms of these orange chromosomes, yielding this karyotype. Again, when we combine these translocations in a double heterozygous individual, the meiosis will again yield a hexavalent. And if between these partially homologous regions, which are flanked on either side by different chromatin constitution, if here a crossover occurs in that region, then in one step we get a new karyotype which did not exist before and is composed of 3 chromosome components, this one, and this one, and co-segregates with the other 2 translocated chromosomes. And at the same time, the wild-type constitution is reconstituted, which does not occur in this individual at all, because this chromosome, and the newly restored wild-type chromosome, this occur in the progeny. A special case is the so-called chromotripsis, which is a multi-break event with multiple misrepair. Here I show you schematically 9 chromosomes of an original karyotype with a lot of breakage and stitching together all the colored region of these 9 chromosomes into a new giant chromosome. If this chromosome is viable and the cells can divide after, for instance, inactivation of this centromere, then either a cancer may be developed or a new species with a strong sexual barrier is on the way of origin. And that such events can really happen. We could prove in the Witzer-Faber again by this configuration, which is very seldom one. And you see that 1, 2, 3, 4, 5 of the 12 chromosomes are linked into a single multiple translocation event. And this does not only occur immediately in an experiment, we have also indirect evidence that it occurs in nature. For instance, the duckweed genus Spirodela consists of 2 sister species, very similar morphology, namely Spirodela polyritza, which has 20 chromosome pairs, and Spirodela intermedia, which has 18 chromosome pairs. When we take a bug library, which is specific for each of the Spirodela polyritza chromosome, and hybridize them on the karyotype of Spirodela intermedia, we observe that 8 chromosome pairs of both species seem to be structurally similar and not rearranged. However, chromosomes 20 and 15 of polyritza make a reciprocal translocations forming chromosome 18 and 16 of intermedia. Similarly, chromosome these 2 of polyritza undergo a reciprocal translocation into chromosome 12 and 11 of intermedia. And chromosome 8 and 18 of polyritza make a translocation combining them into a metacentric combined chromosome number 9, by this way reducing the chromosome number from 40 to 38. And here we have a very complex rearrangement involving 6 chromosomes of polyritza and yielding finally 5 chromosomes of intermedia, reducing the karyotype to 36 chromosomes only. And it is very unlikely that these events occur step by step. Subsequently, most likely at least these and maybe all rearrangement are the result of one multiple breakage and misrepair event. So further on, I want to show you that also chromosome number, size, and shape of chromosomes of a karyotype, which vary via DNA misrepair, have an impact on genome stability, namely very low chromosome numbers, which occur via descending disploidy, restrict, of course, the meiotic segregation and the number of crossovers. Oppositely, very high chromosome number, which can evolve via ascending disploidy or via whole genome duplication, they increase the risk of missegregation. Of course, they have more crossovers, but the risk of missegregation is higher. Finally, chromosome arms, which are longer than half of the spindle axis of the average mitotic spindle, are unstable. This is what we have shown in the late 90s and demonstrated at the Faber-Beam. I will give you some details in the next slide. We prolonged one Visa-Faber chromosome by secondary rearrangement with this translocated chromosome in one step, and then here with another crossover and with this chromosome in the next step. The consequence is that this arm is now longer than half of the average spindle axis, and when the cell wall is formed, it cuts through these elongated chromatids, as you see here in the microscopic photo. The result is that the deleted regions form micronuclei, and this leads to the cell apoptosis. Here you see the micronucleus in vivo. The longer the chromosome arms exceed the average spindle axis, the more frequent such events occur, up to the point where too few cells from meristems can form differentiated tissue, and the entire system is collapsing and the seedling is dying. So this we showed first in Faber-Beam, later also in Barley, and by other colleagues it was later shown to occur in mouse and in yeast, and it is most probably also occurring in Drosophila. So these were the first 3 points, but also very small chromosomes are unstable, but the bottleneck for them is not mitotic division, it is meiotic division, and they are unstable due to reduced meiotic pairing in metaphase 1, as you see here in the small Visa-Faber chromosome, which forms univalence instead of pairing, and in anaphase, where the cohesion of sister chromatids is insufficient, and this yields into micronuclei and often in essential deletions, which makes the gametes unviable. So, but the tolerance of small chromosomes is very species specific. Birds, for instance, can tolerate much smaller chromosomes than, for instance, wheat, pizza fava, or barley. And even if the bottleneck for small chromosomes is meiosis, if you in tissue culture make the chromosomes even smaller, then also they are not cohesed enough during mitotic divisions and get lost also during mitosis. So furthermore, acrocentric or telocentric chromosomes experience less crossovers, but they also need less double-strand breaks for diploid rearrangements than symmetric meta or submetacentric chromosomes. So on this slide, I want to show you that also DNA double-strand break misrepair can cause local duplications, gene duplications, sequence duplications. For instance, if a break happens and the break ends are resected and forming overhangs, which then get replicated into the donor double helix, and the resynthesis goes farther than the breakpoints was, and then the ends meet via microhomology, and the gaps in the recipient chromosomes are resynthesized, then we get a duplication of regions in this new recipient chromosomes. And indeed, we could show in the case of Itza-Faber that very long, up to 100 megabases, hundreds of kilobases long sequences can form during gene conversion, which can be made visible in the microscope due to labeling with space analogs. And that after homology-dependent repair, an end joining of these ends may occur, has been shown in barley, also in a work with our group together with Max Planck Institute in Cologne a couple of years ago. So then I have here a warning for genomic people. This warning refers to the so-called non-reciprocal translocation. The non-reciprocal translocation in the sense of the word should not occur. It means that there is, as here shown, a loss of heterozygosity in terminal chromosomal region. How can they occur? One way is so-called break-induced replication repair, and results in conversion from the break end up to the end according to the donor sequence. This mechanism is experimentally proven only in yeast. It is postulated to occur in humans, but there is no molecular evidence so far provided. And in plants, we were searching for such events in the large genomes. In faba bean, we could not detect such event. And even in the smaller Arabidopsis, the group of Charles White could not find indication of BIR. So it is rather likely that what we call non-reciprocal translocation is the result of unbalanced segregation of a reciprocal translocation event. So usually when you have a chromatid translocation, then in the first mitosis after its origin, it forms such a kind of quadrivalent structure. And if this undamaged chromatid and this wild-type chromatid segregate to one pole, and both of the translocated chromatid segregate to the other pole, we have here a wild-type daughter cell and here a balanced segregation. If, however, the arrangement is like this configuration, then it would happen that a wild-type, that a wild-type chromatid and a translocated chromatid segregate to one pole, and the other wild type chromatid and the other translocated to the other. And then you get something which looks like a non-reciprocal translocation, which is a terminal loss of heterozygosity. So finally, I come to the strategies of genome size evolution, which are again linked to misrepair of double-strand breaks. So we postulate the strategy number 1, which results in a shrinkage due to the fact that more deletions or larger deletions occur than insertions. And this deletion biased repair would yield smaller chromosomes and genomes. But there is a risk. The smaller the genomes become, the higher is the risk that the next deletion will involve an essential gene, and this would be a dead end. This is overcome in evolution by whole genome size duplication, which gives an update for the next round of deletion. And indeed, we observe whole genome duplication much more frequent in groups in groups of organisms which have small genomes, as Brassicaceae, for instance. But recurrent whole genome duplication leads to an exponential increase of chromosome numbers, which in turn increase the risk of missegregation. And interestingly, in the same group of living organisms, we very frequently find diploid reduction of chromosome number, which yields a diploidization of this duplicated genome, again linked with deletions. So the second strategy is chromosome or genome expansion. In this case, the insertion is faster and more frequent and more longer sequence involving than deletions into a break. So the expansion can again go up to the point where energetically the replication and the transmission of the genome becomes difficult. So the third strategy we call stasis. In this case, the frequency and the length of insertion and deletions are more or less equal and do not change the chromosome size much. So we have indeed found that Arabidopsis thaliana in experimental analysis of a single double-strand break reveals that even today there is a deletion bias DSP repair, so more deletions than insertions are caused in the Arabidopsis genome. However, if we do the same experiment with the medium-sized barley genome of 5 gigabases, we found a stasis. So the deletions and the insertions are nearly in balance. And we postulate that the large genomes somewhere occurred during the strategy number 2. And of course, it is possible, sometimes by mutation of a single component of the repair complex, to switch from shrinkage to stasis or vice versa, or from expansion to stasis or vice versa. And this assumption can also explain the so-called C-value paradox, which tells us that the genome size is not directly correlated with the genetic complexity of the carrier organism. So therefore, we consider ourselves as very complex organisms, and many plants have a many-fold larger genome than humans. So with this, I come to summarize my talk. Double-strand breaks can be lethal if they are unrepaired or if they cause essential deletions. They may result in ontogenetic differentiation by chromatid elimination or immune globulin gene rearrangement, a topic which is important for animals, but I could not touch it today due to time restrictions. Double-strand breaks can change the chromosome and the genome size via deletions or insertions, including retrotranspositions. They can rearrange the linkage via conversion or crossover, or the chromosome structure via inversions, conversions, I should say, via inversions or translocations. They can rearrange the chromosome size up to the higher or lower tolerance limits. They can alter the chromosome number via ascending or descending disploidy. They can lead to cancerogenesis or to speciation via primary or secondary rearrangements, and in particular via the special case of chromotripsis, which means multiple breakage and rearrangement. Therefore, we have to conclude correct double-strand break repair maintains genome stability. Un- or misrepair double-strand breaks are usually deleterious. However, repair which alters the pre-break situation is the main source of genetic novelty. When it causes tumors, it is deleterious, but if it leads to adaptation and speciation, it is evolutionarily beneficial. So at this point, I thank you for your attention and again for inviting me, and I would be ready to take questions, but maybe you could write them down because my hearing insufficiency does not guarantee that I understand your questions correctly. Thank you very much.
Thank you so much, Professor Schubert. What a wonderful talk. We literally walked through double-strand breaks and chromosome debris and visualized how through the many possibilities that emerge, we come out with the prospect of evolutionary changes brought on by chromosome and genome size modifications. So, of course, we would have loved to hear you go on and on, but there's time's restriction. So there's a question in the box, sir, if you can go down to the chat box. Shamrat has posed a question.
Can
I read it to you?
Chat box. I try to open. Are there specific chromosomal regions that are more susceptible to epigenetic modification during speciation? Yeah. I should answer in two parts. The first part is the chromosomal rearrangements are definitely clustered. They can occur randomly everywhere, but they survive mostly when they are inside or at the border of heterochromatin, because in this case, the risk that they destroy essential genes is pretty low. Whether they also are more susceptible for epigenetic modification, I do not think so, because the susceptibility for epigenetic modification is generally given by chromatin structure. And finally, we are not so sure whether epigenetic changes don't go back to genetic changes. In many cases where, for instance, the group of Ales Petchinka was investigating this, the epigenetic changes in a karyotype have a forerunner in a genetic change. So, but of course, this is not necessarily exclusive.
I hope you have your answer, Shamrat. So there's another question.
Genome size modification is mainly owing to rearrangement and is in a general notion, so this can be taken Yeah, it is my belief. Of course, the point mutations which alter gene expression or the structure of encoded gene is important. But for the evolution of species, double-strand break-based rearrangement are much more important, and because they also cause a genetic barrier because of the heterozygous depression of fertility. And only if they become homozygous, the fertility is normalized in most of the cases. Therefore, we believe that for speciation, the genome rearrangements are the most important events.
I hope you have your answer, Biplab. So thank you for your questions. Can I now request Professor Anita Mukherjee, Vice President of the ARCHANA Sharma Foundation of Calcutta, to Biplab wants to ask another question. So would you take it?
Yeah, of course.
Of course. Okay, shoot, Biplab.
Hello, am I audible?
Yeah, hello.
Hello.
Biplab, please write it. It would be easier.
How does the cell decide about the choice of repair pathway? This is a difficult question because, for instance, homology-dependent repair mainly occurs after replication, but not exclusively. It can also occur before, while non-homologous end joining occurs throughout the cell cycle. And then we have some preliminary indications that the sequence can decide whether homologous recombination or another pathway is chosen. And also the composition of the repair complex has an impact. So it's not a simple answer on that, but a combination of several ones. And of course, when you have a break in a repetitive sequence, the chance that other repetitive sequences are nearby increases the chance of homologous recombination. Furthermore, the SMC5-6 complex can in G2 after replication, after breakage of chromosome, mediate that homologous region come close to each other. How this works via cohesion is not 100% clear, but it can happen. So I see another question. Faba bean were mutated for the chromosome translocation. Pardon, I did not read the paper. Aha, yeah. In Faba bean, the story started in the 50s, where my former supervisor and his colleagues generated a lot of individual translocations in seedlings of Vizza faba. Then they grow these seedlings up and wait until the translocations were transmitted heterozygosly to next generation. Then they self this heterozygous generation and selected homozygous translocations. And then these homozygous translocation carriers were crossed again, and the F1 was selfed again to combine them and to allow these different types of experiments which I have you shown. Is that clear or?
Yes, sir, it's clear. I think he's got the point. So can we move on to Professor Anita Mukherjee for her vote of thanks and final words?
Yeah, here is I see just another question. somatic cells prefer NHA, is that to increase the chance of mutation. As I mentioned, it is not true that non-homologous end joining is error-prone and homology-dependent repair is correct. Both of them can be either correct or error-prone. And the lesions, the double-strand breaks, are usually by chance. But what happens via recombination, this can be sometimes non-random, as I mentioned. This is this type of the pain in zero phase. I do not understand well the question of Chandan Day.
He's probably asking whether this type of aberrations can be repaired in the G0 phase.
Also, an aberration cannot be repaired at all. The break can be repaired, but the aberration is no longer recognized by the cell as a mistake. It cannot be repaired at all. It can be transmitted or can be extinct if it is lethal, but it cannot be repaired.
We are not taking any more questions. I'm sorry. If you have further questions, you can probably leave them. We can forward them to Professor Schubert later, but we have to go on with the last part of the program.
Okay.
So I'm requesting Professor Anita Mukherjee to please come up here and continue the program.
Thank you very much. This was a pleasant duty of mine to give this vote of thanks on behalf of the ARCHANA SHARMA Foundation of Calcutta. At the onset, we are indebted to you, Professor Schubert, for giving us this opportunity, and you accepted our invitation to give this lecture in memory of our revered teachers, Professor ARCHANA Sharma and Arun Sharma. We are all very grateful to you, and indeed, it was a mesmerizing lecture through which we went. And this is as a formal, formality of this oration, part of a formality. We offer a token of appreciation from our behalf. And this is the certificate we would like to give. And I hope you will accept it. And at the same time, one of our students, Sritama Bhadra, I think you have met her before. She's online also, and she will be visiting us in January, and so we are planning to express our gratitude in some other ways also, and I am sure you will accept that. And it's also a request on behalf of the ARCHANA SHARMA Foundation. Thank you very much, and I would also like to thank the University of Calcutta, Department of Botany, and the faculty for giving us this logistic help. And moreover, the head of the department and her team had done a tremendous job to help us organizing this oration. Thank you very much.
Thank you very much, too.
So after that wonderful lecture, we will take leave for the day. Thank you so much, all the online and offline participants. It's been a real pleasure listening to Professor Ingo Schubert, and his lecture was literally just out of this world. And it has left me itching to go back to the textbooks once more. Thank you so much, Professor, and thank you everyone for your attention and full-hearted participation. Thank you very much. Oh, I just want to do one more thing. I want to thank Mr. Akash Mondal and Braindrops for giving us the logistic support and organizing this lecture. Thank you so much, Mr. Mondal, to you and your team. Sir, you are saying something, Professor Schubert.
Thank you very much. It was a great pleasure and honor for me. And I hope we can keep in contact and exchange scientific ideas in future.
Thank you, Professor Schubert. We would be very happy if you could manage to come here, because we are going to have a conference, international conference in December, but as it is not possible for you to visit that time, so we have this lecture now, and I do hope that we can have you again sometime in near future. Thank you very much.
Thank you.
Thank you.
Thank you.
Oh, bye-bye.
উনি যাওয়া অব্দি কাটাটা ভালো না